All of the information presented in this video is sourced from our textbook, the video on replication in the lecture slides, and the lecture slides. So DNA replication. Once replication begins, so we have our double-stranded DNA. It starts at a origin of replication, which is where the replication fork starts to unwind the DNA, and it keeps moving along the DNA. DNA helicase attaches to the fork, so let's say the fork is right here, and it breaks the H bonds that are holding the double strands together and makes single strands. So single-stranded binding protein binds to each single-stranded DNA and protects it from anything that could happen to it, while gyrase at the same time binds and relieves tension from the unwinding. So at that point, RNA primase synthesizes primers to allow DNA synthesis to begin. They're just kind of synthesized down here. Then DNA polymerase 3 adds nucleotides one by one and is required for this DNA synthesis. So it does this in the, oh shoot, you're supposed to be broken apart. I'm sorry, I forgot to tell you. The helicase broke you apart. Anyway, so it does this in a leading strand and a lagging strand. So since they're anti-parallel, the leading strand from 5' to 3' is one solid DNA synthesis, but the lagging strand up here is going to be chopped up and make Okazaki fragments. It's made from the discontinuous DNA synthesis of the lagging strand. Then DNA polymerase comes in and cuts out the RNA primer, and ligase seals the nicks from the DNA polymerase and the primer without actually adding any more nucleotides. So that, in short, is DNA replication. Thank you. In replication, we also have a template strand, which is used by the DNA RNA polymerase, which attaches complementary bases during replication. It goes from 5' to 3', and then the coding strand is the DNA strand whose base sequence corresponds to the base sequence of the RNA transcript produced. There are some of the stuff that was in the word box that was not required for replication, but required for transcription. Synthesis of primers come from RNTPs, and RNA polymerase 3 helps transcription in eukaryotes instead. Also, if DNA polymerases were not able to perform their functions, they would not be able to recognize the correct places to bind and would not be able to recognize a lagging strand or a leading strand, so Okazaki fragments would kind of get lost.